Welcome to the NMI podcast, and today we're speaking with Dr. Denise Furness. And before we get into the conversation, which will largely revolve around the topic of nutrigenomics, I'll give you a bit of background into Dr. Furness and her academic career as well as clinical career and so on. So she is a pioneer in the field of nutrigenomics and personalized health with over 20 years experience in the area. She began her research career in 2003 investigating folate and B vitamin metabolism, methylation, and DNA damage in relationship to pregnancy health. And over the years, her research expanded to include things like vitamin D, immune health, oxidative stress, as well as both male and female fertility. And a lot of this research we're talking about was conducted at a premier institute globally, especially in the area of nutrigenomics. That's the University of Adelaide's CSIRO Human Nutrition and the Women's and Children's Hospital in Adelaide, Australia. During this time, she witnessed, as part of that research, a really high success rate in recurrent miscarriage and high-risk pregnancy clinics. She's published her work from this, all this involvement in peer-reviewed journals, has won several awards for her research, and has been all around the world presenting this work and continues to do so at our conferences and so on. In 2012, she founded Your Genes in Nutrition and began working with integrative doctors and learning more about integrative medicine and functional medicine. And since then, she's really become an integral part of this field, I would argue. She's regularly lectured for the Australasian College of Nutritional and Environmental Medicine, that's ACNEM. She's editor of the ACNEM Journal and on the editorial board of the Nutritional Medicine Journal. And she regularly lectures globally at conferences for health professionals on topics related to nutrigenomics and functional medicine. On top of all of this, she runs a busy clinic and works with a range of patients, helping them identify underlying triggers and root causes of their health concerns using this functional or integrative approach, applying genetics alongside biochemical markers, as well as nutrition and lifestyle medicine as the mainstay of intervention. So no small introduction, but it's, with that background, I think listeners can agree it's a great privilege to be able to speak to you, Denise. So it's great to have you on the podcast.
Thank you for that wonderful introduction. Yes, there has been a long history, and I still have three kind of big things going on in the sense of still running the clinic, seeing patients, doing a lot of education, and then a lot of advisory roles in relation to nutrigenomics and epigenetics, because this whole world of personalized health is just growing. So I feel very fortunate to be in this space.
Yeah, absolutely. And it's great that we have you in this space. You're a great ambassador for the area. One of the questions I wanted to ask before we get into, what nutrigenomics is, what it means for people in clinical practice and so on is how you got into this area. Firstly, as an academic. So
I almost want to say accidentally, but I guess it wasn't accidentally because I was looking. So I was already working as a geneticist, or back then we called a molecular biologist. So my major was molecular biology, and I was working at CSIRO, but not human nutrition, not doing the nutrigenomics. I was actually looking for genetic variations, so SNPs, the same things we do for nutrigenomics, but in relation to viruses and trying to determine the genetic variations that allowed a virus to jump from, say, an animal to a human. And these were paramyxoviruses, not coronaviruses, but the same kind of thing that the world knows about now. So I was in that virology space, loved the genetics, loved the laboratory I was in. It was amazing, PC4 lab, really well funded. Thought I'd found my dream job. But as time went on, I started doing some animal work, and I just wasn't okay with that. It just doesn't sit well with me. So I was fine in the lab and, doing all the experiments and stuff, but the animal side of things just really affected me. So I started looking around at other areas in genetics and saw this opportunity, a PhD. So at the time I was just working as a research assistant, done my degree and my honors. And I saw, Michael Fennick and his nutrigenomics work and DNA damage. And I was like, oh my goodness, wow, I didn't even know that these gene variations could influence our dietary requirements, our ability to absorb nutrients, this DNA diet concept. And as someone who was genuinely interested in health, I tried to eat well and exercise, very interested in that space. I was totally drawn to it. The only disadvantage was that I was in Victoria. Michael's lab was in Adelaide, but it was well worth the move. I was lucky enough to get a scholarship, so went and joined his team and did my PhD there. But yeah, so that's how I found it. I went searching for genetics kind of roles that didn't involve animal work and work with humans.
Yeah, it's amazing. And it's almost like, the complete opposite in the, with animal work, you're killing experimental animals and you went into like bringing people into the world through through your work in pregnancy. It's quite a
Yeah.
contrast. There's some
To be completely honest, the fertility thing wasn't really what drew me in. It was more about understanding I don't think I even really understood what the project was to start with. I was young, 23, 24. I remember thinking I was old at the time though, thinking should I embark on a PhD? But the fertility thing was something that actually I started to understand how important that was later. Like as a younger person, I didn't really understand how common it was for someone to have recurrent miscarriage or pregnancy complications. I was very much interested in more so because we did folate nutrigenomics, MTHFR, MTHFD1, all of those genes. I just wanted to understand how the genes influence dietary requirements. But you are right. We did move into fertility, obstetrics, gynecology. That's where I went, and it was great because I was working with humans that were consenting to having their blood taken or working with you.
yeah, it's a great way to put it. So you've mentioned, and I wanted to ask you about this. You've mentioned your work with Professor Michael Fennick, and I think probably people who are in the field of personalized nutrition and genetics, that name will be up there with one of the most prominent sort of names in the field. His work has really helped put nutrigenomics and nutrigenetics on the map in relationship to personalized nutrition. And it's been a while since I read through his work, but a lot of his work was focused on this area of DNA damage and genomic stability and its relationship to nutrient requirements and redefining RDAs and things like that. So, that's was pretty incredible work. And I'm just wondering, as part of that group, did that thinking around redefining our approach to nutrition through Dr. Fennick's work influence the way you think and the work that you were doing?
Oh, 100%, and still does today. Learning again, if I just come to folate, I mean, it's one nutrient, but he started out actually as a radiation biologist. So he was interested in DNA damage and he got into more that sort of nutrition space. I don't exactly know directly how, but he basically found out that if you're low in folate, you have really high amounts of DNA damage. So he developed a particular DNA damage test. It's called the cytokinesis block micronucleus assay, big name. We just call it CBMN assay. But you take someone's blood and then you culture it and we look for DNA damage. And it's a really robust test. I wish that I could do it with my patients now, but it's not automated. It's a bit more laborious. You've got to, get it, look down the microscope, score the DNA damage. But he moved into that nutrition space and I think was quite shocked himself at the impact that, folate B12 has on DNA damage. And then really moved into that methylation space. And then that expanded into looking at the genetics and how some of these SNPs also influence DNA damage. So that's really where he started and really pioneered all of that early research around what he would call folate and B vitamin metabolism. A lot of the practitioners listening probably call this methylation, but he just called it, folate B vitamin metabolism. As a scientist, when we talk about methylation, I often talk about one carbon metabolism. That's where we focused and how I published it. But now as a practitioner, we sort of say methylation. But he really did open my eyes to the impact that nutrients can have on our DNA, not just looking at the SNPs influencing our dietary requirements, but if you have deficiencies, you get this DNA damage, this cellular damage, and it's profound. You can see it. Like you look down the microscope. You can do it in cell culture work, which I did initially just to learn the assay and learn about this. But then we can see it in our patients. So we would be measuring, levels of folate, levels of homocysteine, levels of vitamin D, and then you're matching the genetics, the DNA damage with these nutrients, with other biomarkers.
Yeah, it's amazing. Yeah, it's interesting the semantics there. Like I've hearing you speak, it sounds like if we just rephrased methylation as B vitamin metabolism, it would solve a lot of the conundrums that we have, like in people who are translating this to clinical practice. But that's probably a whole nother matter. So
We can do a separate podcast just on that.
yeah, I think we will. But to continue with this theme of like just understanding how you arrived where you are now is, you've gone from this, deep sort of academic career basically, and then branched into functional medicine or personalized nutrition, environmental medicine. Like, how did that bridge happen? I'm curious to know.
Yeah, so once I finished my PhD, we had really good results, particularly in relation to DNA damage and seeing higher levels in those that had sort of what we call adverse pregnancy outcomes, preeclampsia. I was then lucky enough to get a postdoctoral fellowship with NHMRC, and I continued my work, not so much with CSIRO, then more so in the hospital, the women's and children's. Even though I already worked with patients from there, I was very much involved in the recurrent miscarriage clinic, and still linked with Adelaide University. But my role, I guess, was a little bit more translational in the sense I was doing less lab work, even though I sort of set up the study. It was the PAPO study. I was still involved in recruiting, but I had a research assistant by then, a PhD student, and I was working very closely with the main obstetrician, Dr. Dean McCormack. She's amazing. And then my main supervisor was Professor Gus Decker, so he was the head of sort of maternal fetal medicine at another hospital. So very clinical and literally in the hospital. And although my background was initially more so science, genetics, molecular biology, biochemistry, I started doing a lot more nutrition just through that sort of PhD in nutrigenomics. So I was doing nutrition reviews and dietary records and, working one-on-one with the patients, and I loved it. So I was like, well, I really like working with the patients. To be honest, I thought about doing medicine. If I hadn't have got sick, which is the next step, I would have sat the GAMSAT and done postgrad med. But I got Graves' disease. I was really, really sick. I had to step out of work for a while. And that experience really led me into integrative and functional medicine. So my own personal diagnosis, in a way, didn't make sense when I first spoke to the endocrinologist. So they couldn't explain why I got it. And I understand that autoimmune conditions are multifactorial. We all know that now. But at the time I was a patient and I didn't know. And basically he said to me, you'll need to be on, this medication and we'll remove your thyroid. There's, you won't recover, et cetera, all these kind of things. And I guess as someone that understands genetics, understood epigenetics, because that's the other thing that we did in our lab. So I used to measure the methyl groups on the DNA. So my area of, I guess, expertise in that space of fertility was epigenetics, the methylation pathway, the genes, the diet. And we had others in the lab that also did telomeres and stuff. So, I really understood cellular health and that, sick cells, damaged cells should be cleared away, apoptosis, necrosis. We used to look at that down the microscope. So, if the body's working well, we'll clear out those damaged cells, those infected cells, or whatever it may be, and we will make new cells. Your DNA replicates, you get every day new skin cells. The liver can repair quite quickly. We repair, we regenerate. So this kind of diagnosis, even though I was happy to get a diagnosis because I was quite sick, I knew that wasn't my prognosis and that I believed things could change if I could work out what my body needed. Again, thinking back to nutrition, it's like there are certain, maybe I've got some deficiencies, which I did. I was quite shocked. I thought I was healthy, but I was definitely burning the candle at both ends before becoming unwell. I was working at a high level, training at a high level, socializing at a high level. All the things as a functional medicine practitioner. We let people know you need a bit of downtime, a bit of nourishment, lacking probably a bit more nourishment, lacking a bit of sleep, high stress. But yeah, just that answer from the endocrinologist in saying that we need to remove your thyroid, quite in intensive treatment, and yeah, it just didn't make sense. So then I moved into integrative medicine. It made sense because, I recovered. Plus, really the success we had in the recurrent miscarriage clinic. We had an 80% success rate. And I didn't realize how good that was until we started speaking at other events and other conferences, and really starting to understand that when you understand your DNA and what your body needs, and it's not just genetics alone, you still need to look at blood biomarkers and understand the person as a whole, but you have the ability to get someone well. And that led to me getting integrative medicine. You mentioned ACNM. I was at an ACNM conference, and I said to the audience, and I'd been presenting at ACNM, which is at that stage, it was just integrative doctors. Now we work with naturopaths and allied health and pharmacists. But I said to the integrative doctors, do you think there is a space for nutrigenomics, DNA testing in clinic? Now, this was 2012, maybe 2011, 2012. And then someone came and grabbed me as I got off stage, because I was presenting some of my research and, talking about nutrigenomics and saying, I think that it should not just be done in a research sense. We should be doing this in clinical practice. And 3 of the sponsors at this conference were DNA testing companies. So I had been in my little bubble, my academic bubble, not realizing that this was already happening in the real world. Because when you're a scientist or an academic, even though you're working in this space and you might believe in your results and your research, you don't realize that other people, those fast adopters are out there.
Yeah.
So that really helped the transition. I met doctors that said, we want you to work in our clinic. I started working with those doctors. At the beginning, I just interpreted, genetic reports, DNA, learnt lots while working in integrative medicine practice, did a bit more study, and here we are.
Wow, that's amazing. That's really interesting. Yeah, I definitely see that disconnect between, the research and clinical practice. And I think that the term you use, early adopters, is really reflective of what a lot of people are doing in functional medicine. Is they're really fast to adopt things. It's interesting. That's a great story. Thanks for sharing that. It adds a lot of depth and insight into how you got where you are now. And I think as what you've really become is like a leading educator on nutrigenomics and so forth. So I thought, you know, in terms of early adopters, one of the challenges we have around that is that a lot of healthcare providers don't have any formal training in nutrigenetics or nutrigenomics, yet these tests are widely available. They're also now direct to consumer as well. So if you're the general public, you can go and get these done and then, maybe drag them into your doctor and start talking about them and so on. So I thought what we could do is like wind it back and just break down what we're talking about even as a starting point, and then get into a little bit more depth. So, what is nutrigenomics? Like, what are we even talking about here broadly? Is this a good place to start?
So just to let the listeners know, in case you're not familiar with the term, hopefully you're not there Googling, nutrigenomics was something that, was a term that definitely we used a decade, 2 decades ago, or nutritional genomics, nutrigenomics. We're basically talking about the interaction between, nutrition, food or nutrients and our DNA, and how that influences our health. So we're not just thinking about the genetics. We're not just thinking about the food. We're thinking about the relationship, that, connection between the two, and how that influences health. As time went on, nutrigenetics, which I've heard you mention a few times, really became the word, because nutrigenomics, technically, you're talking about the whole genome. So nutrigenetics, technically, scientifically, is a more appropriate word, because with nutrigenetics and what we do in nutrigenomics is we're looking at SNPs, which are single nucleotide polymorphisms. They're just one base pair change in the DNA, and that can influence the protein that gene codes for, like the shape. It might not bind to B12 as well, and therefore you're not getting as much B12, drawn into the cell for B12 absorption. So we're looking for these variations, and in particular, those that relate to food. But the area has grown in 20 years. No longer is it just about nutrition. Now we're thinking about environmental impacts as well. So we're looking at chemicals and toxins that influence our genes, stress, other things like that, sleep, exercise. So a better term that's often used these days is functional genomics, tying with functional medicine, or Jeffrey Bland calls it lifestyle genetics. And, talking about Michael Fennick and his research, I think the story when I went to IHS, my first IHS Integrative Healthcare Symposium in New York, Jeffrey Bland, who's, one of the fathers of functional medicine, got up and presented our research from Adelaide, and Michael Fennick. So I feel very lucky to have worked in his lab. He really has paved the way in this space of, genetics and personalized health. So yeah, it's a term that I think will change. At the moment, I'm saying functional genomics, and now that I'm starting to do a lot more teaching, because I think that helps people understand that it's more than just nutrition. It's really that lifestyle and how our environment, our food, our stress impacts our DNA, because some of us not only need more of certain nutrients, we might need a bit more sleep, or we might not tolerate stress so well, or we might be sensitive to some chemicals and toxins. And we can understand that when we understand our DNA.
Yeah, I love it. That's a really interesting perspective of the evolution of what it is, what we're looking at, and how the term is changing as well. Functional genomics just makes a lot of sense. So a term you've mentioned a couple of times is single nucleotide polymorphisms, which are often abbreviated to SNPs. And this is possibly a term that most people listening to this podcast have heard before, but I'm just wondering if you can break down what that actually is. And then I guess the second part of that question is, beyond what a SNP is, like, how does that relate to a personalized nutrition and lifestyle medicine?
Yeah, I would love to tell you that. So your DNA, think of it like your life book, right? These are your instructions. They're coding for not just what you look like. We typically think about, phenotype being your eye color, your hair color, your height. But phenotype means what you're expressing, and that is coded for in our DNA. So it's your instruction manual, your life book, however you want to call it. But it's made up of 4 bases: adenine, thymine, guanine, and cytosine. We call them A, T, C, and G. So when we talk about a SNP, or a single nucleotide polymorphism, we're simply talking about a 1-letter change in that DNA code. So nucleotide just means that one of those letters. We also call it a base, a DNA base. So a single base pair change, a single letter change in the DNA can result in a big change in the protein. So just to explain your DNA, they're the instructions, but they need to be read. They then need to be turned into RNA, which is kind of like the messenger RNA. And then we have transfer RNA, and then you get a protein. And it's the proteins that do the work. The proteins are structural proteins. They're transporters. They're enzymes. They go off and make, hormones, neurotransmitters. So the proteins do the work. They're what's made, and the genes are the instructions for the protein. But sometimes we have these base pair changes in the DNA, the code, and then the protein ends up being a little bit different. Just to give you a little side story, we knew it was coming because that's what always happens with me. If the listeners don't know me, there's always tangents. Ozzy Osbourne has just passed, and they have done his entire genome, and he has a variation on one of the alcohol dehydrogenase genes that they think has actually influenced his ability to tolerate so much alcohol. So here's a great case because they're trying to understand Parkinson's. There's also a gene, this would be more of a mutation. This would be what we call clinical genetics. So just to step back for the listeners to understand genetic testing as a whole, there's different types of genetic testing. So if you are looking for a mutation that causes a rare genetic disease or cancer, that's clinical genetics or medical genetics. And that needs to be done by a medical doctor who has specialized, who's done advanced training, specialist training in genetics. And their job is to diagnose disease based on SNPs, or what we call mutations in that case. And they'll be working with a clinical, genetic counselor. And that genetic counselor is then there to support those patients and explain what that means for them and their family. Often these conditions, these rare genetic disorders can be, shorten people's lives. They're pretty extreme. So the reason I said that is because Ozzy Osbourne happens to have a pretty serious mutation on a gene that they don't exactly know what it does, but they think it's influenced his brain because apparently his brain works a little bit differently to most people. And obviously he is well known for being in the band, etc., but he has dyslexia, he has major learning issues, but has this seriously addictive personality, but also obviously been very successful in his career. So the human genome sequencing is something that can be done, but just for the listeners, I don't recommend it. Don't jump online, and you can get it now for under $1,000. I'm not suggesting you do that, because unless you have someone like the Ozzy Osbourne doctors that wanted to go through it all in detail, no one's going to be able to interpret that for you. And even if you came and saw someone like me, or if you found a nutrigenomics practitioner in the UK, they're not going to have the software, the bioinformatics to understand your genome. So even though there's a lot of whole genome sequencing going on right now, you need to be doing that with clinical geneticists, with people that have the software, people that can interpret it. The type of genetic testing that we're talking about, that's offered through a number of testing companies for functional medicine practitioners, naturopaths, allied health doctors, even pharmacists here in Australia are doing it. And obviously what, we have through pure genomics, if people are doing, testing as well. So that kind of genetic testing, we are not looking to diagnose a disease. We are looking for common variations, what we mentioned, SNPs, getting back to your original question, long story, but we're getting there. SNPs, these single variations influence dietary requirements like MTHFR with folate. Now, folate doesn't cause a disease. MTHFR, doesn't cause a disease. It doesn't cause miscarriage. It doesn't cause depression, but it does have a significant impact on folate and homocysteine and that methylation pathway. And depending on the person and their diet and their environment, that could manifest into something. But it's that diet, gene, environment interaction. So that's what we're doing with nutrigenomics or functional genomics. We're looking for genes that influence your susceptibility or your dietary requirements. And then what we do when we have that information is we give that patient or client information to kind of compensate. Do you need a little bit more of folate or B12? Or do you need some more vitamin D? Or do we really, you've got your comped AA, which is not me, I'm a fast comp, but you're a slow comped. You might be a bit more susceptible to stress. It's really important that we put in some stress management tools for you while you're dealing with these serious things, because we know that you don't break down your adrenaline, your stress hormone as quickly as someone else. So you're helping people understand their bodies, why they might feel or act a certain way, and personalizing their nutrition information, personalizing that lifestyle intervention to optimize their health. Long way to answer, sorry.
No, it's good. It's excellent to have that context and that depth in the answer, I think, and also for people to understand the layers and of nutrigenomic or genetic testing and where those different sort of scopes of practice fall as well, so that I think really useful context. So really appreciate that. I guess a question that wouldn't mind digging into around the SNPs also, as you've mentioned that the SNP itself doesn't cause disease, but we do hear this sort of dialogue around genetic testing is that there are bad genes and good genes, and you're unlucky if you have this and that. And I'm just wondering if we can dig into that a bit, is like, are these genes bad or good, or, are they there for a reason, or, why do we have these variations if they hinder our health, for example?
Yeah, I love this question. I feel like this is what I'm put on earth to do, amongst many other things, but is to help practitioners and patients understand that there isn't something wrong with them. And unfortunately, there has been, I think, this miscommunication along the lines of nutrigenomics, and that some genes are bad, like they're really, and people can start to think there's something wrong with them. And the reality is, in most cases, and we even have this information, we don't even have to hypothesize, but in most cases, these SNPs that we think are a negative actually have had some kind of advantage in the past as we've evolved. And honestly, in all of the SNPs I think about that we classify as having a negative effect, or that we want to try to work with to optimize health, it's not the gene, it's the environment. So if I just talk about MTHFR, and I want to make it clear that's not the most important gene. There are so many other important ones: ApoE, BDNF, COMT, TCN2 with B12, FUT2 gut. I could just go on and on and on. But the reason I talk about MTHFR, one is obviously it was something that I used to look at in my research. So I've been working in this space for over 20 years. But also, the people listening to this have likely heard about MTHFR because there has been so much information online. So to use that as an example, MTHFR, there are two SNPs, 677 and 1298. The 677 variant has a bigger impact than the 1298. And just to explain what I mean by those numbers, when we talk about 677, we're talking about the location of the SNP in that gene. So we've got sort of all of our nucleotides, 1, 2, 3, 4, 5, blah, blah, blah. So we've got the whole gene and at position 677. There can be a C or a T. A C is classified as so-called normal, or you're not likely to have that sort of negative impact. And when it comes to the 1298, which is a little bit further down in the gene, it can be an A or a C. With MTHFR, particularly those that have a 677TT, we know that's associated with approximately 60, potentially even a 70% reduction in enzyme activity. So what does that mean? MTHFR converts one form of folate to another. It's actually called 5,10-methylene, is the sort of the precursor folate, into 5-methyl. That's exactly what MTHFR does. It has one function and it's one direction. So it goes from one form of folate to the next. When MTHFR is slowed down because people have these SNPs, we get less of that methyl folate. Now, that can be an issue because methylfolate is needed to convert something called homocysteine into methionine. So what methylfolate does is literally stick a methyl group onto homocysteine, and that methyl group added to homocysteine is methionine. That's then needed to make what we call S-adenosyl methionine, or really just the methyl groups that are needed for all the methylation reactions in the body. So if you're low in methylfolate, you tend to get high homocysteine. That can cause vascular damage. That can cause, resulting methylation issues. You're not getting those methyl groups being made. So there can be issues, but it's not the MTHFR, particularly not on its own. If you have plenty of folate and B2, which is the cofactor for MTHFR, you may not. And plenty of people with MTHFR SNPs don't have high homocysteine, don't have these methylation issues. Plenty of people with MTHFR have had babies and haven't had miscarriage, even though there is an association. But again, the association is linked with lower folate environmental factors. So with MTHFR, what we do know, though, is that the SNP is actually, the SNP that we think is a so-called negative, is actually associated with a reduction in malaria. So it helps protect against malaria. In addition, when you've got more of the other folate, so when you slow down this 5,10-methylene to 5-methyl, 5,10-methylene actually is a form of folate that goes off and supports another part of the pathway for DNA synthesis and repair. And we have evidence to show that that SNP, although a negative, can be protective against skin cancer in those that are in areas with high UV, because obviously you want to repair that DNA damage. So you're going to get more of that 5,10-methylene. So it's likely at some point, because the SNP is so common, that it was a huge advantage, whether it was because it was protecting people against malaria, helping those, with DNA repair. But the reality is our environment has changed so much. And once upon a time, there was plenty of folate. Folate's in green leafy vegetables, it's in nuts, it's in seeds, it's in citrus. But people aren't eating whole food anymore. Or if they are eating broccoli, which is high in folate, it's sat in the supermarket for a few days. It's heat and light sensitive. And then they've put in a pot and they've overcooked it and all the folate's broken down. So even those that are trying to eat well are often not getting the same level of nutrients that they once had. So they're not getting the folate in. In addition, one last thing I want to say about methylation, because MTHFR and folate and B6, B12, these all support something called methylation. In this modern world, we have a much bigger demand, I would use that term, sort of demand on that methylation pathway. And I say this because methylation is also involved in detoxification. So if you are exposed to certain chemicals and toxins, mercury, arsenic, if those things are around, methylation's needed to clear them. When you are under high stress, which most people are under high stress in this day and age, methylation is needed to get rid of the adrenaline. There are a lot of endocrine disruptors in our environment impacting estrogen. Methylation is needed for estrogen clearance. So you have this higher need for folate and Bs, and then you're not getting as much from your diet because of the way that we now process our food. And then you have a SNP, and now it becomes a big problem.
Yeah. Yeah. No, that's a really fantastic example. And I think it really points to the nuance in this. It's not that you can just run a genetic test, identify a good or bad gene, and then prescribe a supplement and hope that, that's going to be the antidote or the solution to personalized nutrition. It's really this global view of how this individual functions within their environment and what those, external things are that are have a relationship to their genetics. So I think that leads us into another question I really wanted to ask is how, how does this testing, potentially benefit personalized recommendations? And you've touched on a few things, and I think it'd be good to bring this back to like a more functional approach in that it's more than just food. So you've mentioned food, but there's also environment, and I believe there's also some work on behavior, both motivational behavior, but also behavior of the individual in relationship to their genes. So what I feels like is emerging as a pretty holistic, for want of a better word, view of genetics, in that it can inform quite a bit around personalized recommendations. So can you share how SNPs can guide us, or if they can?
Yeah, I 100% think SNPs can guide us. I mean, that is what I do day to day with my patients. It's what I teach. But like what you said, it's not as simple. It would be easy if it was just a SNP. Here you go, have this supplement, do this. We still need to think about the person as a whole, because with someone, they might have a SNP, but they might not need a supplement because actually their diet's great. They're not under high stress, whatever it may be. Whereas someone else, in actual fact, let's give an example of vitamin A. So there's a SNP called BCMO1, beta carotene monooxygenase 1. It converts beta carotene into retinol that then makes retinol for vitamin A. So we know that people that have these SNPs in BCMO1, we call them poor converters. They don't convert the beta carotene, so that's coming from the plant sources of vitamin A, your oranges and reds and yellows. They don't convert that beta carotene into the retinol so well. Now, the reality is you would think, well, if you've got the SNP, you're going to need more retinol. We need to talk about, animal sources, or we might need to supplement. Do they have the symptoms, vision issues, pimples, etc.? But on occasion, you might actually get someone who genetically does not have the SNP. Genetically, they're not looking like they have a high requirement for retinol, but their diet is so poor, or they're not absorbing their fats because they've got a major digestive disorder, or, maybe their stools are just, they're getting rid of all the fat. They've got floaty stools, whatever it may be. They still might need more vitamin A. So the genetics are very important. They guide me. But I also, like, I'm sure all the practitioners, listening, if they are practitioners, it might be a mixed audience, but practitioners will often have, I've got a symptom questionnaire, you've got your history. So you're matching the genes with the person. What are their major health concerns? What are their symptoms? Are the genetics the explanation? And often they are. They might tell you that they have palpitations when they drink coffee, and you're like, well, you're a slow caffeine metabolizer. We can see you don't tolerate it. You're at higher risk of palpitations, getting anxiety. Most often the genes will explain it, but occasionally the genes won't. Maybe there is something else going on. So we always want to be pairing genetic testing with either some blood biomarkers, or at least, symptoms and questionnaires, and finding out what's going on in that individual's, unique makeup. So yes, the genes can guide us, but they are not a standalone. So it's very different to say that medical genetics. And the difference, I guess, between nutrigenomics or functional genomics and clinical medical genetics is when you have that rare SNP, it doesn't matter what your diet or your lifestyle is. You've got that gene, you've got that disease or that risk. With nutrigenomics, you might have the gene, but it really comes down to your environment and your diet, or you might not have the gene, but the reality is you can still have issues if you are really deficient or you are exposed to a high levels of chemicals and toxins. Does that kind of make sense? I mean, I don't want to overcomplicate it, but at the same time, I think it's such a useful tool, but it needs to be considered in association with that person, their goals, and what's going on in their body at that moment in time.
Yeah, it makes perfect sense, I think. And something that just struck me too, I'd never really thought of before, but it appears to me like learning about these genes and the way they function more broadly can teach you quite a bit about the function and physiology of an individual, regardless of whether or not the genes are variant, which is not something I'd really thought of, but that's cool.
It's so cool because you're not only learning as a practitioner, your patient all of a sudden understands at such a deeper level, because the kind of person that invests in this testing, they're genuinely interested in their health. They want to know what's best for them. And you're starting to talk about, the chemicals and toxins in the environment or the foods and how it affects your DNA. And all of a sudden, they look at food differently too. It's not just calories in and out and, macros and protein and this and that. They're like, oh my God, these micronutrients, this B12, this vitamin D. The vitamin D interacts with my genes and it turns on, cytokines to support my immune system. Of course, we don't get into too technical terminology like cytokines, but I tell people that vitamin D is interacting with your genes and regulating this whole sort of support network for your immune system. And that's why vitamin D is so important. But the other thing with genetics too, and I think you sort of touched on this briefly before, as a practitioner, I guess one of the difficult things is getting your patient or your client to follow that protocol. And of course they want to, but the reality is the biggest issue in today's world is we're all busy. We're busy, we're stretched, we've got so many things going on. We might forget to take our supplements, or we are, not doing that walk. We're not making time to, cook these healthy meals, and we fall back into old habits. Once you understand your DNA, you're no longer just being told, this is healthy for you. Just go and do this. It's deeper than that. And I guess one thing we haven't touched on too, even though it's not clinical genetics, we do often talk about, chronic conditions, whether you're more at risk. And of course, it's not a diagnosis, but are you more at risk of an autoimmune disease? For example, I've got the top 3 risk factors: HLA, human leukocyte antigens, for Graves' disease. And I got Graves' disease. Once I understood my genetics, once I understood everything involved, it kind of took a lot of the guilt and shame and everything away, because you're like, oh, it was bound to happen. I was deficient in vitamin D. This is the lifestyle I was leading, and I've got these genes. So it actually, I didn't do anything wrong. I'm not going to blame the universe. That's my story. But for someone else, it might be they've got issues around regulating their blood sugars, and they might be at high risk of type 2 diabetes. So we can talk to them and say, these are the genes that you've inherited. Likely they've got a strong family history as well. But once they understand they've really got those genes, we can start to talk to them. And there's no DNA diet, but we can give them guidelines and help reduce that risk. For me too, with ApoE4 and Alzheimer's, this is an area I'm very passionate about. And there's so much we can do to reduce risk of Alzheimer's. And there's a lot that we can do to find out our risk factors. So for those that are interested, we can get so much information about ourselves and then use that to optimize our diet and lifestyle. And you don't have to be perfect. We're all about having fun. That's important for brain health and overall health as well. But making some tweaks and understanding what's going to be good for you, and also where your kind of playing field is. I'm Australian. There is a big alcohol drinking culture in Australia. I like having a drink, quite a social person. For me, understanding my genetics now, and I'm very focused on healthy aging, reducing my risk of Alzheimer's. I mean, my mum was having symptoms at my age, during perimenopause and menopause. I've cut alcohol, not completely. If I want to have the odd drink, I will. I'm all about enjoying yourself. But I now know that that's not something that is good for my brain, and I've made a conscious decision. So the genetics can help you make these decisions. And to be honest, they just make it easier. You just go, you know what? What's more important?
Yeah.
I'm going to do this. And it takes the difficulty away from following what's probably a simple protocol or a simple intervention, but often in life we're just too busy to pay attention to these simple things.
Yeah. Yeah, it's there's something about genetics, isn't it? And there's quite a, for listeners too, there's quite a bit of data to back up this idea that genetic testing is an incredibly powerful motivator for behavioral change. You run a genetic panel, that you personalize the diet a bit, and then, they're much more likely to follow it than without genetic-led counseling. But one of the things you are sort of speaking to, I think, too, is that there's something different about genetics versus like a blood biomarker, which maybe because it's more variable, or maybe because genetics sort of almost speak to what it is to be human and our ancestors and our hereditary, and it's fixed. There's just something different about genetics versus other tests, basically. Yeah.
I think you actually said it right there. Those blood biomarkers will and can change. That homocysteine can go up and down. Your cholesterol can go up and down. Whatever your iron levels could go up and down, but your genetics is fixed. You can't change that. You've inherited them from your parents. It's not that they're bad. We all have genetic strengths and susceptibilities. Once upon a time, some of those susceptibilities were probably strengths, but in today's society of reduced nutrients, Western processed food, high stress, blue light, our life, the amount of chemicals and toxins, our body has to work differently now to stay well. And we have to make some of these conscious decisions. And if you understand your genetics, you can then tweak things to help you live your best life in this high-paced, chemical-laden processed food world. And
Yeah.
of course,
Yeah.
most of your listeners probably are well aware of those issues and probably already making steps to reduce their chemical load, to eat well. But having this information, I think, can sometimes just make little differences that I see firsthand with my patients' work. And as you said, the amount of data, we even have good randomized controlled trials showing for long-term change and benefits. DNA testing really outperforms being told by someone you've got high blood pressure, lower your salt. But if you've got high blood pressure and you've got SNPs in ACE and AGT, so that angiotensin converting enzyme, you're at risk of high blood pressure, and these genes interact with salt, you're likely to manage your high blood pressure with lower salt. We see that 12 months later, the people that did DNA testing stay off salt, whereas the ones that didn't do DNA testing, despite being told to stay off salt, they might have done that. Most people do make change initially, but then they go back to old habits after a short amount of time. Whereas with genetics, I guess the big thing is, even in the UK, they did a study comparing with your gold standard diabetes dietary intervention. So they compared DNA testing, giving personalized advice against the gold standard diabetes study. I think her name's Justine Horn. She is a dietitian, but specializes in nutrigenomics and works in the academic space. She's published a lot in this area, but she showed that nutrigenomic advice actually provided much better results in helping people manage their diabetes compared to the UK gold standard, lifestyle diet intervention.
Yeah, it's a great example. Um, as we're going to have to move through this quickly, but I really wanted to speak about the BioAge study that you're doing. So, for listeners, Denise has been running I guess you might call it a pilot trial through her clinical practice around the use of genetic testing, personalized recommendations, and also measures of biological aging, which includes epigenetics. So I'm wondering then if you can give us an overview of what that study's all about, and then we can dive into what you're seeing as in terms of results.
Yeah, and you may even want to do another podcast on this. And as I said, we'll share the results with NMI, and you can publish this as well. So this started in 2023. For those listening, epigenetics and biological age has become, really popular in the last few years. And up until recently, it was something that we couldn't do as a commercial test unless you had a lab. So 20 years ago, 15 years ago, I had a lab. I used to do epigenetic testing and genetics. but we haven't had a test that we can do as practitioners, whereas now we have these epigenetic methylation tests, and we know that these link with what we call biological age. So chronological age is your birthday, you know, how long you've been on the planet. We can't change that. But biological age can change, and it's more to do with your cellular health. So you can have accelerated aging and be older biologically compared to your chronological age, or you could be younger. So when these tests came out in Australia, I launched a pilot study or a real world study. We had 60 people sign up. We had just over 40 finish properly. Some went overseas, some didn't finish the protocol, some did the test a little bit later, but we didn't include them in that 3-month intervention. And what we found is that with a whole food diet, so 6 serves of veggies, 2 serves of greens, 2 serves of cruciferous, 2 serves of colored, being, you know, purple, red, orange, yellow, and relatively high protein. So we had an exercise physiologist who guided them for exercise, 3 strength training sessions a week, depending on their level. That might be quite basic if they hadn't done a lot of strength training. And then just half an hour of movement, you know, every other day. That could be a walk, just making sure they're moving every day for about half an hour. And we also had movement snacks if they're people like me that sit all day. So with the diet, the exercise, an app. We had 5 supplements. Am I allowed to say what the supplements are?
Yeah, go for it.
No. Yeah, so we had 5 key supplements. Now, I wanted to have about 20, of course, but I try not to give my patients too many supplements. So we went through a B complex. So the B complex plus by Pure Encapsulations, and I really like that because it's sort of 400 micrograms of folate. It's not too high. Methylation can be a bit sensitive. We don't want too much, too little. Got the methylcobalamin, and then the cofactors you need for methylation. So the B6, the B2, bit of, you know, B3, nicotinamide, etc. So we had that. We had resveratrol. We had the liver GI plus, which has got some more nutrigenomic things like sulforaphane, bit of NAC. So sulforaphane, which comes from cruciferous vegetables, literally goes in and talks to your DNA and turns on antioxidant enzymes. So that was a great one. And then we had a little bit of nicotinamide riboside, which I know you can't get in the UK just yet. We were very limited here in Australia. We only had 100 milligrams. A lot of the studies suggest you need a bit more than that. And basically with this supplement protocol, diet, lifestyle, we saw massive improvements in some of the biomarkers. Now, not all of them. Telomeres is something we tested, though most testing interventions don't see a lot with telomeres, but we saw a reduction in biological age. In the 3 months, I think the Levine's pheno age was about 2.6 years in 3 months, statistically significant. But the most exciting thing for me, who's all into DNA damage, who understands that DNA damage is kind of the foundation of health, if you listen to Michael Fennick, we saw biological age using a clock called DAM age, which is published in Nature Genetics, and Stephen Horvath and others have put that together. We saw a reduction. Now I've got to be careful, is that the 6-month data or the 3, but it's 7 or 8 years. I think it might be the 6 month we got to 8 years, but about a 7-year, like huge, statistically significant reduction in biological age that's associated with DNA damage and cellular damage just through eating a whole food diet, reducing obviously processed foods and sugars. We did fortnightly sessions during that 3 months, teaching people about chemicals and toxins and endocrine disruptors, trying to reduce them in their environment. And we had, for anyone that's into biological aging and knows Brian Johnson, who has, you know, his stuff. If you look at the results we have and some of the results he gets, we're getting things on par. You know, we're slowing our biological age with Dunedin pace. Even myself, my results to the equivalent of what he's doing with these $2 to $3 million interventions. So it just shows you through diet and lifestyle how much of an improvement you can have, and not only what you can have, but in actually a relatively short amount of time, you know, only 3 months. Mind you, everyone was very compliant for that first 3 months. You know, we're in a group program, we're all chatting with each other, but it shows you it's possible. It also taught me how dynamic these methylation patterns are in our DNA, because I'm a bit more old school. When I learned about methylation more than 20 years ago, we learned that it's really robust, almost fixed. You know, we knew it could change, But I would say in the last 20 years, we've realized just how much it changes. And you can think of epigenetics, methylation, as almost a quick way to adapt to your environment. So genetics like SNPs, these variations have evolved over time to help us navigate our environment. They were a benefit at some point, otherwise they wouldn't be common. And look at where we are in the food chain, we're the top species. These SNPs were likely a benefit. But epigenetics can change very quickly. So if there is a stress, a chemical or a toxin, a sound, a smell, whatever it may be, our environment influences our epigenetics, and it can change very quickly. And it can change in a positive way, which is associated with reduced biological age, or it can go the other way and it can change in a way that actually is associated with accelerated aging. And we did see that. We had one person who had a tumor in his ear, and during the study had gamma radiation, and his biological age went all the way up. It's likely that's very transient. It doesn't stick. Biological age is dynamic, but it can go either way.
Yeah, it's a great example. And thanks for the overview of the study. It's a lot to pack into, 10 minutes of, explanation. I think you're right. We probably need to unpack this another time, but it's an incredible example of like really where the rubber meets the road for genetics is you've integrating like this the testing of single nucleotide polymorphisms with integrative medicine and personalized care and lifestyle changes. And then I think the proof and, the cherry on the cake with this is the using these biological aging assessments to really look at the overall impact of this type of care or intervention on health outcomes in a way that, as you say, we never really could before. And I think it's worthwhile highlighting too, just for the prosperity, I think, is that you were recently in London and by chance, arguably, you presented this alongside Stephen Horvath. You were you followed his lecture, him presenting the technical work that he'd done to bring this, into the science. And you had stepped up and presented like, this is how we're using it in clinical practice. So how was that experience?
Oh, I feel so blessed, and it was phenomenal. And I think not only was I excited to meet him, he was really impressed with the data. And there are some results that I didn't show. I've talked to you about, some of the main findings, but one of the tests that he thinks is really important, or the test that we should be doing, is called Grim Age. Now, I don't do Grim Age. Grim Age is really time till death. It is named after the Grim Reaper. As a practitioner, I don't know how even though if you get benefits, it's great. As I said, I'm all about empowering people, not putting the fear in them. I'm very cautious with my language. So the Grim Age, I actually have a meeting on Thursday in 2 days with the Clock Foundation, which is his foundation, which is exciting, which has come off the back of that talk.
Hmm.
But he was explaining to me how the Grim Age is probably the best one for really showing an improvement, or, extending your kind of what we call health span. So not just, longevity, but healthy years. And I told him that with the 3-month intervention, we saw a 6-month reduction in Grim Age. So all up, that's kind of 9 months, because they would have aged 3 months. And then we saw a little bit extra in the 6 months, not as much. Just to qualify that, people in the 6 month didn't follow the protocol as strictly. Some experimented with bringing alcohol back in. It was approaching Christmas time.
Yeah.
But he blew me away. He said, that's fantastic. He said, we're doing all this research. He said, we're not even seeing a 6-month reduction in 3 months. He said, that's a really short time in his world. He said, that's very impressive. So I was just on a high. I can't wait to meet with the Clock Foundation and talk about this in a lot more detail. And yeah, I think he was excited as a scientist to see that we're actually using the research that he has developed in a clinical setting, but also then to see the interventions and what we can do just from a lifestyle perspective. Because even though I would say he's the world leader in biological age clocks and epigenetics, I think what I gathered from him is his knowledge around nutrition, and nutrition being able to really improve cellular health, reduce biological age. I think that for him was actually a little bit striking.
Yeah, that's really interesting. And I think that's one of the wonderful things about cross-pollinating research with people who are practicing, integrative or functional medicine, needs to be more of that. That disconnect needs to close, because what people are seeing in clinical practice does look miraculous often, when you're applying these therapies. So yeah, I think unfortunately we're going to need to wrap up. It feels like we could go on for days, but thank you for your time. There's some really rich content there. I will highlight to listeners some of your work that'll be published in the notes against the podcast, and we'll also publish a link to your website. And I'll mention also that you are running a program that healthcare professionals can engage with if they want to learn more about nutrigenomics. What was the name of your course or
It
program?
is the Academy for Precision Health, and we are focusing on nutrigenomics or functional genomics as I teach them, epigenetics, and really personalized health. So, as I mentioned to you about trying to bring it all together, for those of you who are functional medicine practitioners, even touching on some of the other common tests we do, stool analysis, Dutch, and how that would support the genetics and the epigenetics. So, yeah, I launched that at the beginning of this year. First cohort of 30 practitioners, and it's really exciting. We've got the second cohort coming soon. So,
That's
yeah,
amazing.
I absolutely love it. I love it. So, yeah, we're shaping the future of precision health together.
Yeah, absolutely. And, we really appreciate the work you're doing. Just on behalf of the audience, thank you for your time. And, thank you for all the work that you put into this over decades now, and, haven't lost any vitality or enthusiasm for it. It's palpable. So looking forward to seeing what the future holds, and we'll reconnect again for sure.
Thank you so much. I'll talk to you soon. Bye.